Hey everyone, and welcome to This Explainer. Today, we're taking on a genuinely fascinating mission. We're going to explore exactly how a medical laboratory solves one of the most high-stakes biological mysteries happening inside your very own body right now, identifying your blood type.
Picture this number in your head for a second. 45%. That is the estimated percentage of people worldwide who walk around every day with Group O blood. But how do we actually know that? How does a lab definitively prove this invisible, microscopic fact about your biology? A fact that, quite frankly, could literally be the difference between a totally successful treatment and a massive medical emergency. Okay, let's dive into this. Globally, Group O leads the pack at 45%, followed pretty closely by Group A at 40%. Then we drop down to Group B at 10%, and finally, the super rare Group AB at just 5%. Now, these aren't just fun statistics. They represent the real-world global stakes of blood typing. Before anyone can safely receive a transfusion, a lab technician has to navigate these exact odds using incredibly precise procedures, and that is exactly what we're investigating today. Here is our roadmap for today's detective story. First, the ABO blood system. Second, the antigen-antibody reaction. Third, essential lab reagents. Fourth, slide and tube methods. Fifth, forward versus reverse grouping. And finally, grading and troubleshooting.
All right, part 1, the ABO blood system. Think of blood biology like a giant, strict lock-and-key system. Right on the surface of your red blood cells, you have these specific markers called antigens. These are your locks. Group A has the A antigen, Group B has B, AB has both, and O basically has neither. Now, floating around in the liquid plasma are antibodies, and these are the keys, specifically designed to seek out foreign locks. So Group A blood has anti-B antibodies in the plasma, primed to defend against what it doesn't already have. Group O, on the other hand, naturally possesses both anti-A and anti-B.
Moving right along to part 2, the antigen-antibody reaction.
So the crucial point is this fundamental biological law known as Landsteiner's rule. It states that if a specific antigen is present on your red blood cells, its corresponding antibody absolutely must be absent in the plasma. Why? Because if this rule were broken, your blood would literally attack itself, causing a visual clumping called hemagglutination. This one simple rule is the entire foundation for all lab testing we're about to see.
Part 3, essential lab reagents. Because labs handle massive volumes of these critical tests, commercial antisera are globally color-coded. It's a brilliant system designed to prevent fatal errors. Anti-A is always blue, using a patent blue dye. Anti-B is always yellow, thanks to an acroflavin dye. And anti-D, which determines your positive or negative Rh factor, that one is kept strictly clear. So you essentially have vital safety mechanisms built right into the chemistry itself. Number 4, slide and tube methods. When you get into a practical lab setting, there are really two primary battlegrounds for testing. On one side, you've got the slide method. This is a rapid, qualitative screening tool. It's perfect for bedside testing or those intense emergency situations where you need an answer on the fly. On the other side, we have the tube method. This is the absolute gold standard used in dedicated blood banks. It's highly sensitive, incredibly precise, and it uses centrifugation to force reactions that might otherwise be way too weak to see. With the rapid slide method, the pacing is fast. You place 3 separate drops of the patient's blood on a glass slide. Then, you add the anti-A, anti-B, and anti-D separately to each drop. You mix them with completely separate wooden sticks, gotta avoid that cross-contamination, right? And then rock the slide gently. But here is a critical real-world lab tip. You must read that result within 2 minutes. If you wait any longer, the edges of the liquid start drying out, which actually mimics the look of real clumping and creates a disastrous false positive.
Now, for maximum precision, labs switch over to the gold standard tube method. First, technicians prepare a precise 5% red blood cell suspension in normal saline. They mix the suspension with the antisera in small test tubes and pop it into a centrifuge at 1,000 RPMs for 1 minute. This aggressive spinning physically forces the antigens and antibodies into intense contact, forming a tight little pellet or button of cells at the very bottom. The true skill of the lab tech comes in the very next step. You have to gently shake the tube just enough to dislodge that cell button without breaking apart any of the fragile clumps that might have formed. It's a really delicate balance.
All right, part 5, forward versus reverse grouping. And this brilliantly illustrates a core rule of medical labs. You never, ever rely on just one test. Forward grouping targets the antigens directly on the patient's red blood cells, using those known color-coded antisera we talked about. That's your primary identification. But, to be absolutely certain, the lab performs reverse grouping. They basically flip the script. They test the patient's liquid plasma to hunt for the antibodies they expect to find, using known lab-provided reagent A and B cells.
To put that into perspective, let's say we add anti-A to the patient's blood and it clumps. Boom! We know the A antigen is present. That's the forward test. But in reverse, we expose the patient's serum to those known reagent A and B cells. If the patient genuinely is A-type, their plasma naturally contains anti-B antibodies, which means it will only clump the reagent B cells. The primary identification and the confirmation flawlessly mirror one another.
Finally, step 6, grading and troubleshooting.
Now, what's really interesting about this slide is that labs don't just lazily jot down positive or negative. They rigorously grade the reaction strength on a scale from 0 to 4+. A 4+ reaction? That is undeniable. It's a massive, solid aggregate of cells that leaves the surrounding liquid completely clear. A 1+ reaction, though, might actually require a microscope to spot the tiny, fine clumps. And a 0 grade is just a totally smooth, reddish suspension. Every single grade tells the scientist exactly how strong that biological interaction truly was.
But let's be real, science isn't always perfect. Sometimes labs face a real biological puzzle known as a discrepancy. This happens when the forward and reverse tests mysteriously contradict each other. Imagine the cells react like group A, but the serum shows anti-A antibodies. That completely breaks Landsteiner's rule. Now, this could just be a simple technical error, like a bad label or dirty glassware, but it could also be biological. For example, very young infants haven't fully developed their antibodies yet. Or maybe the patient has a weak genetic phenotype that hides from standard testing, like the weak D variant. The lab detective has to systematically troubleshoot all of this just to release a safe, accurate result.
And that brings us to the end of our explainer. Identifying your blood type really is an absolute masterclass in meticulous biological detective work. From colorful reagents to high-speed centrifuges and mirrored verification tests, these technicians leave absolutely nothing to chance. But it definitely leaves you wondering, right? If something as simple as waiting 2 minutes too long on a glass slide, or a microscopic weakness in an infant's plasma can throw off our entire understanding of who you are at a cellular level, what other hidden microscopic rules are silently dictating the survival of your body every single day? Thank you so much for exploring this invisible universe flowing through your veins with me. Stay curious, and I'll catch you in the next one.